Hi, I'm Kelly Mukai, and over the summer, with the help of Dr. Han at iRepertoire, I researched the effect of estrogen level fluctuation throughout the menstrual cycle on my immune repertoire, or diversity of T cells. I first would like to thank Mr. K for allowing me to do this research and for supporting me throughout my learning adventure. I wouldn't have been able to do it without him. My research question for my project was, is my immune system weaker when I am menstruating, or am I more prone to becoming sick when I am on my period? I wondered this because I often find myself becoming sick when I am menstruating versus when I am not. I was very interested in this topic because as a teenager, I wanted to learn more about how hormones affect my body. The immune system is what protects us from becoming infected by a variety of pathogens. There are many components to the immune system, but the immune system can be broadly divided into two parts: innate immunity and adaptive immunity. Innate immunity, or nonspecific immunity, is the body's first line of defense. It will kill any unwanted pathogen, but does not differentiate between one pathogen and another. However, for this research, I specifically looked at adaptive immunity, or acquired immune system. Adaptive immunity is antigen-specific, which makes it more efficient because of its ability to differentiate between a variety of pathogens. The adaptive immune system contains B cells and T cells, but for my experiment, I specifically looked at T cells. T cells have antigen-specific receptors which generate the diversity of the immune system. The T cell receptors are differentiated by the VDJ genes, which are randomly inserted and rearranged. This rearrangement occurs in the thymus, which is the gland next to the heart, where T cells mature and develop. When the VDJ genes are rearranged, they form the unique CDR3 region, which is what has antigen specificity and is the binding site for those antigens. An average person has 5 times 10 to the 11 T cells, majority of them being in tissue. However, the unique number of T cells depends on the person's health condition. Our immune repertoire is a very important indicator of health. Each little colored square is a unique antigen-specific CDR3 region. The size of the square depends on the frequency of unique CDR3s. This is why the left repertoire has a low diversity, making that repertoire not as healthy as the right repertoire, which has many unique CDR3s. When the diversity is lower, you are more vulnerable to getting infected by a pathogen because there are less unique antigen-specific CDR3s to protect you. Throughout the menstrual cycle, estrogen levels are constantly fluctuating. In this graph, you can see that on day 1, when menstruating, estrogen levels are extremely low compared to at ovulation when there is that estrogen spike. These fluctuating levels have a direct effect on the number of T cells present in the body. Prior research has already shown that estrogen increases the amount of T cells present in the blood. Estrogen is a lipophilic steroid which allows it to diffuse through the cell membrane and affect the genetic material directly in the cell. As you can see in this diagram, estrogen is able to enter the cell to aid in gene expression. Have you ever wondered why women often outlive men and have a longer life expectancy rate by almost 5 years? Well, this is because females have a more responsive and robust immune system compared to males because of their sex hormone estrogen. This is why for my research, I specifically answered the question, does estrogen have an effect on my immune repertoire? This research is important because no one has looked at this before. Most researchers have only looked at the effect of estrogen on the number of T cells present. I will be the first person to use iRepertoire technology to look at the effect that estrogen has on the diversity of T cells present, which is a very important indicator of health. I hypothesize that my diversity of T cells will increase at ovulation when there are higher levels of estrogen. Therefore, because estrogen levels are low when I begin menstruation, I hypothesize that the diversity of T cells will be lower at that time. To have a quantitative measurement, I looked at the D50 value. The D50 value looks at the degree of diversity of T cells within a sample. This D50 value can be calculated by the number of unique CDR3s that make up 50% of the total reads multiplied by 100 divided by the total number of unique CDR3s. The CDR3 region again being what determines the antigen specificity of the T cell. Therefore, because of this equation, the closer the number is to 50, the more diverse the T cells are and the healthier I am. Figuring out when to take my test was a very long process. It required me to use multiple LH and clear blue ovulation strips to determine when I would be ovulating. I used LH strips, the pink strip on the bottom, because when LH strips are positive, it means ovulation will occur soon. When the left line began to get darker, I began to use the clear blue digital advanced ovulation kit, which has a 99% success rate at determining the day of ovulation by tracking both LH levels and estrogen levels. When a happy face appeared on the screen, it meant that I was at peak fertility or ovulation. Throughout that entire time, I had kept a food log and exercise and sleep schedule as constants for my experiment. After I was able to figure out when to take my test, I had to learn how to take my tests. This process was very stressful. In order to use iRepertoire technology, I needed to prick my fingers with this orange tool and get enough blood out of the finger to fill 4 filter paper circles. I began by washing my hands thoroughly with soap and water. I then wiped my finger down with the alcohol wipe and had to press the tool to puncture a hole in my finger. I squeezed the blood into those 4 circles and used gauze and band-aids to stop the bleeding. Anything that got into contact with blood was safely disposed of in biohazard bags. After I sent the blood samples to iRepertoire Hudson Alpha in Alabama, it went through a long process to finally be able to see the diversity of T cells in that sample. First, RNA was extracted from the dry blood spots using the RNeasy microkit. Then, using reverse transcriptase, the RNA was converted into complementary DNA strands. iRepertoire multiplex primer sets were then used to amplify the CDR3 region of the T cell receptor chain. Then, using next-generation sequencing, which is designed to sequence millions of DNA molecules simultaneously, we were able to see the effect that estrogen level fluctuation had on the diversity of T cells. This is a reminder of what my hypothesis for this experiment was. I hypothesized that my diversity of T cells will increase at ovulation when there are higher levels of estrogen. Therefore, because when estrogen levels are low and I begin menstruation, I hypothesized that the diversity of T cells will be lower at that time. Fortunately, my results agreed with my hypothesis. When looking at menstruation versus ovulation, the diversity of T cells were lower for both me and Kayla when we were menstruating. Because of the way Kayla's and my experiments overlapped, I was able to get 4 tests when I was ovulating and menstruating to confirm my results. This lower diversity means that I have less amounts of unique T cells that can attack foreign antigens when I am menstruating, leaving me more vulnerable to becoming sick when I am on my period. I was also able to average the D50 values of the 4 tests I got per menstruation and ovulation. And the test results show that when ovulating, there's a higher diversity of T cells than when menstruating. The blue blocks are my D50 values, and the green blocks are Kayla's D50 values. There was a pretty significant change in the D50 values from menstruation to ovulation for both me and Kayla, which allowed me to see that the diversity of T cells most likely has a direct relationship with the levels of estrogen present. These results definitely supported my hypothesis. The phenomenon of why I often become sick when I am menstruating was supported by this experiment. This experiment can give information to those women who often find themselves becoming sick when they are on their period like me. It also shows to women the importance of taking care of their bodies throughout this vulnerable time. Other experiments that could further my research would be to repeat my experiment again using other people as their test subjects. The reason for similarity in results for Kayla and me could be because we are genetically identical twins. However, I do know that there are other people who find themselves becoming sick when they are on their period. So if you are another person with that problem, know that you are not alone. This experience has taught me so much, and if you have the opportunity, I really do recommend you to take advantage of it and do research. Some advice that I have is to not be afraid to research what you are interested in. It's a bit awkward to talk about menstruation, but I was interested in how hormones affect the body. It will definitely make your experience so much better if you find a topic that you are genuinely intrigued by. Another piece of advice is to get things done earlier in the summer. This prevents you from having to rush as much toward the end. It definitely is hard to give up part of your summer to research, but trust me, it's worth it. Last but definitely not least, remember to have fun with your project. You will learn so much more if your mind is open and if you really want to learn more about the topic that you are researching. I'd like to thank Dr. Han and Dr. Pan from iRepertoire for teaching me so much about how complex the immune system really is and for being able to turn those 4 dried blood spots into beautiful data that showed my diversity of T cells. I would also like to thank Dr. Chan for your constant support throughout this whole experience and for making it very fun to do research. These are my references, and thank you for watching.